Hey everybody, we're going to take just a minute and look at our sarcomere model. So this model is designed to show you a little teensy tiny piece of one of the myofibrils within a muscle cell. So this is a sarcomere. It's actually one full sarcomere, and then many of these sarcomeres would be attached end to end to make up a myofibril. From the outside, we're seeing a representation of a T-tubule in yellow, and then this sort of blue-green color is supposed to be the sarcoplasmic reticulum, which is the modified endoplasmic reticulum responsible for storing calcium ions, which get released just prior to muscle contraction. All right, so I'm going to remove that portion so we can take a look at the actual filaments that make up this sarcomere. So this is a very snazzy model. We can actually loosen it and slide the filaments past one another, which is what occurs during muscle contraction. So during muscle contraction, the thin filaments, which are shown here mostly in red, are going to be drawn inwards so that they overlap our thick filaments, shown here in blue, a little more fully. During relaxation, those thin filaments are released, and the sarcomere goes back to its original size and position. So let's look at the parts of our sarcomere for starters. On the end, we have the Z line. That Z line is composed of proteins that anchor all of our thin filaments together. And we can see a Z line on either border of our sarcomere. In the middle, we have the M line, conveniently named M for middle. This M line is going to be composed of some proteins that anchor all of our thick filaments together, the thick filaments being blue. There are some zones within, or there's a zone within our sarcomere here, and that exists from here to here. We call this the H zone. This H zone is a lighter area in the center of our A band, which spans from here to here. I want you to remember A band because dark has an A in it, and this is the dark stripe within our skeletal muscle. It's dark because of the thick filaments. So that A band never changes size. It is always the same length as the thick filaments. However, this H zone here can decrease during contraction as the thin filaments are pulled in, and is going to increase, get larger during relaxation as those thin filaments slide back to their original positions. So we have an A band again from one end of our thick filament to the other end of our thick filament. We have an H zone, which just has thick filaments in it. And then we have an I band here, which just contains thin filaments. Thin filaments are, as their name implies, thinner than thick filaments, which means they're going to, the area where you see thin filaments is going to appear lighter. So they make the light stripe or striation. When you look at skeletal muscle under the microscope, you get a light striation and then a dark striation and then a light striation. So this is actually one half of your I band, I for light. There's an I in light. The other half would belong to the sarcomere who lives next door. Let's get a closer look at our filaments now. So we're going to focus on our thin filaments, the ones that are primarily red. So each of those small red bead-like things is a protein of actin, and all those actin proteins are going to link together to make up the majority of that thin filament. The yellow filament is called tropomyosin, and it's a blocking filament. That particular protein is in place to block the binding sites on actin, so that actin, my thin filament, can't connect with my thick filament down here in blue, which is made up of primarily myosin. Now these green ones are really interesting. These are called troponin proteins, and these proteins bind to calcium. So when a muscle is stimulated, a nerve impulse causes the release of calcium from that sarcoplasmic reticulum. Calcium then binds with troponin, changes its shape, and pulls this tropomyosin, the yellow protein, out of the blocking position. And once it's out of the blocking position, myosin and actin can connect, and the myosin head will pull the actin filament inwards, shortening the overall sarcomere. And this happens from both ends. It happens from this end as well as this end simultaneously, and it results in overall sarcomere shortening. Now, if all the sarcomeres are shortening together, that means that the overall muscle cell will shorten, and that's going to cause the overall muscle organ to shorten as well.